Looking at the results of a specimen stain, we listed the most likely organisms present in the specimen. Now let's describe the basic process of microbiology testing for identification and susceptibility. Let's take a look at the process of microbiology testing when a patient who's suspected to have an infection presents. So when the patient presents with symptoms and signs of an infection, we'll call this time 0. And typically, it will take anywhere between 1 to 3 hours for clinicians to examine the patient and, if necessary, collect specimens. And these specimens will be from the site of infection. For example, it could be blood or urine or any other specimen. And then the specimen will be sent to the lab, to the microbiology lab for testing. And what happens at the microbiology lab is that you typically have to incubate these specimens in order for bacteria to grow before we can detect anything. And this can be done using automated systems, or it can be done on an actual plate. So these plates are called agar plates. And what they are is that they essentially have nutrients in them for bacteria to grow, and you just let them sit there and incubate under the right temperature for bacteria to grow. And then after a while, typically anywhere between a few hours to a few days, depending on the organism, we may start to find colonies of the organism. Now, of course, it's possible that nothing may grow. So, sometimes it will be difficult to identify. But if there is a specific organism that will be identified, it will be growing typically between 12 hours to 5 days. And depending on what type of infection we are suspecting in the patient, if we think that it might be an organism that's a very slow grower, it will be wise to keep the specimen for beyond 5 days in order to give it enough time to grow. Now, if something does grow, that's when, typically they start to stain it and then look at it under the microscope. So while the process of staining, is not time consuming, now if you look at the entire process, it typically takes somewhere between, half a day to a day, if not more, before they can actually look at it, stain it, and identify, if it's bacterial infection, whether it's gram-positive, gram-negative, or atypical, and then look under microscope to look at morphology. And then there are a series of tests that will be done in the micro lab in order to identify. So this is where they do those, the lactase test and oxidase test and hemolysis test. And so there will be numerous tests that will be done if applicable, in order to actually identify what the exact organism is. And that typically takes anywhere between 1 to 2 days after staining. So now, this is typically day 2 since that patient presented at the minimum. And once an organism is identified, so for example, if they say this is E. coli, then the next step would be to identify antimicrobial susceptibility. So then, it typically takes another 24 to 48 hours to do various testing, and we will go over some of these susceptibility testing. But the key point here is that it will take, another day or two after the organism is identified in order to find susceptibility results. And what that means is that, they will tell you like which antibiotics will be active against this organism and which ones will not be active, so that we can streamline treatment. Now, as technology advances, we have a rapid diagnostic tests that can expedite this process. So, when we actually grow an organism, we can directly put the some of these colonies into rapid diagnostic tests, and these rapid diagnostic tests will be able to very quickly, in a matter of anywhere between under an hour to just 2 or 3 hours, tell us exactly what the organism is and what the susceptibility results are. Now the problem is these diagnostic tests are limited to only a a select few organisms, so they're not comprehensive of every possibility. So it's not a replacement for the traditional process. So even though we used a rapid diagnostic test, we still need to do the traditional identification and susceptibility testing, because in case the patient has an organism that is not included in this rapid diagnostic test, we still need to figure it out, and you don't want to slow down this process while the rapid diagnostic test is processing. But certainly these rapid diagnostic tests are advancing very quickly year after year. Now, what happens in parallel to this process is what we do to treat the patient, because we're not going to wait for this process to complete and then start treating the patient, because the patient could die by day 3 when we find out exactly what antibiotic will work. So what we do is initially we do empiric antimicrobial therapy. So what empiric means is that we don't really know what's causing the infection, but we will do our best guess to, based on the signs and symptoms, we can anticipate what organisms are possible, and we can use antibiotics broad enough to cover the possibilities. So now we can kind of look at the timeline of treatment. So we typically, we want to avoid treatment as soon as the patient arrives, because if you give the antibiotics immediately, you might not be able to identify anything because you kill all the organisms. So you want to get your samples first if possible, and then start treatment after you get the samples. And then, once we get rapid diagnostic results, that's your first opportunity to optimize treatment. So based on what you find, you may be able to change the antibiotics to have something that's better for the patient. The same with, when you have gram stain results, you can change the antibiotics. And ultimately, when antimicrobial susceptibility results are available, at that point we will switch from empiric therapy to pathogen directed therapy, meaning that we know exactly what is the best antibiotic for the patient that will be active against the organism and will be the narrowest that would get to the site of infection and will result in the optimal outcome for the patient. When it comes to phenotypic susceptibility testing, the concept of MIC is extremely important. So MIC stands for minimum inhibitory concentration, and it is the lowest concentration of an antibiotic that inhibits visible growth of a microorganism. And that's why, we say a phenotype is what actually appears. So, it will be visible to the actual human eyes. And that's to distinguish it from genotypic susceptibility testing. So this is, regardless of what resistant genes may be present in an organism, the minimum inhibitory concentration just, shows you what's the minimum concentration to inhibit growth of organism. And there are different ways to measure MIC. So the gold standard is the broth microdilution, and by gold standard I mean that this would be the reference method, and any other method would be something to as a surrogate for the broth microdilution. And here's how broth microdilution works. So you can kind of imagine a series of test tubes in the microbiology lab, and then in each test tube we will have a broth. So what broth is essentially nutrients for bacteria to growth. Now these are sterile, so there are no bacteria in them, and what we do is that we put antibiotic of choice that we are trying to test in these test tubes, and we increase the concentration of antibiotic in each test tube, so kind of double the dilution. So essentially one of them will not have any antibiotics, so the concentration would be 0, and then we kind of double the concentration in each test tube, and then we inoculate these test tubes with the organism that we are trying to test, and we let it grow. And then after the incubation period is over, we can see that it actually, the growth becomes visible. So you can see that it has become cloudy, or the color may have changed. And then you can see at what concentration nothing grew. So for example, in this example, you can see that at the concentration of 64 and above, nothing was growing. So the minimum concentration was needed to kill the organism in this example is 64, because a concentration of 32, the organism was growing in the test tube, but at 64 or higher, nothing was growing in the test tube. And that's how they measure the minimum inhibitory concentration for a specific antibiotic. And MICs, so every antibiotic has a different MIC for each organism that it covers. So if you have antibiotic A, it will have MIC for E. coli, let's say. It will also have a different MIC for Klebsiella, and so forth, if the antibiotic A is active against these organisms. And then the same if when you go to a different antibiotic, you have to measure the MIC for that different antibiotic for each organism. Now, another method to do is disk diffusion, also referred to as Kirby-Bauer method. And the way this works is that essentially, instead of test tubes, which you can imagine that it will be very labor intensive, so broth microdilution takes a lot of space and requires a lot of labor, so it may not be feasible. And that's why we say it's a reference method. So a much more feasible method would be disk diffusion, and here's how it works. So instead of test tubes, you have an agar plate. So again, this plate is sterile. What's on the plate is nutrients for bacteria to grow. And what we do is that you inoculate the agar plate with the organism that we're trying to test, and then immediately after we inoculate the plate, we place several discs. So these discs are essentially different antibiotics. So each disc has a certain concentration of the antibiotic. And when you place these discs in the region near each disc, this antibiotic starts to diffuse. And the further you go from the center of the disc, the concentration drops. Okay, and then at some point when you go too far from that disc, that specific antibiotic may not be present. So each disc has a different antibiotics. So you are simultaneously testing multiple antibiotics for this particular organism. And then if you have multiple organisms, then you will have one plate for each organism. So you can have several plates set up. And then you let it grow. So after the incubation period, you kind of can see that there is essentially a lawn of the organism that grows. And if the organism is susceptible to that antibiotic, there will be a zone of inhibition. In other words, because antibiotics have diffused in this region, the organism will not be growing. And if it's resistant to it, you can see that the organism will grow through the disc. Essentially means that antibiotic was not active. And then we can kind of correlate this to some, whether it's, is it really susceptible, or is there some sort of intermediate resistance going on? So we can measure the zone of inhibition. So you can see this is a circle. They can actually measure what's the diameter of this zone, because as you can see, some of them have a smaller diameter, others have larger diameter. So the larger the diameter means the more active the antibiotic is. And we'll talk about how to interpret this zone of inhibition shortly. And of course, if it goes through the disc, that means that the organism is completely resistant to that antibiotic. So that's disk diffusion. And then another method, disk diffusion only tells you if it's susceptible, intermediate, or resistant. It doesn't really give you MIC. Okay, so broth microdilution will give you MIC, and then based on the MIC, we can decide if an organism is susceptible, intermediate, or resistant to the given antibiotic. This diffusion only tells you if it's susceptible, intermediate, or resistant without the MIC. So another way to do it in order to get the MIC is the E-test. So the way E-test works is similar to disk diffusion. The difference is that instead of placing discs and then measuring the zone of inhibition, you put a test strip. So there is a strip that has a given antibiotic at different concentrations. So you can see that, at the top, it has a very high concentration of antibiotics, and the more you go down the strip, the concentration drops. And then, again, you inoculate the plate, you place the strip, and you let it incubate. And then after incubation, you can kind of see a zone of inhibition, and then you can see at what concentration the antibiotic stopped inhibiting growth. So that would be your MIC. So in this example, we can see that it stopped growing at concentration of 0.126 or higher. So the MIC would be that number, 0.126. So that's something that's also done in practice. And another way that's more commonly used is the automated machine. So there are certain machines that are automated, essentially. So you can see the one example in this picture where you essentially load this with your sample. You can test a lot of samples simultaneously. So there are these cartridges that you put in, and you just run the machine, and it may take some time for the machine to run. And then, in a matter of an hour or so, and then it will actually give you MICs. So then you can, get your MIC and interpret it as susceptible, intermediate, or resistant. And this is, these automated systems are most commonly used for majority of the samples. Now, the issue is that because these machines are difficult to make, they typically come preloaded with certain antibiotics. So if a new antibiotic comes to the market, it may not be available in this machine, and that's where e-test comes in. So anytime a new antibiotic comes to the market, the manufacturers will make e-tests available so that micro labs can test for susceptibility so they can use the new antibiotic. Now, when you get your MIC results, or if you get results from this diffusion, what they really tell you is, those MICs are essentially in a test tube or on a plate, which we call in vitro, meaning it's not inside a living host. Although we know there is a minimum inhibitory concentration, that doesn't necessarily mean that we would be able to achieve that concentration inside the patient's body. So, it has to do with, the dosing of antibiotics that are safe. Would we be able to achieve that concentration in the patient's body? So in order to interpret that MICs and see if it's feasible to use that antibiotic, there are criteria. So just knowing the MIC doesn't really tell you whether you can use it or not until you interpret it. And the way we interpret it is that we use CLSI is the organization that gives us the interpretive criteria or breakpoints for MICs. And and I will show you how to navigate this document. But essentially, they will tell you for a given organism, for each antibiotic, they will tell you at what MIC cutoffs it will be considered susceptible, intermediate, resistant, and for some of these there will be a susceptible dose dependent, meaning that it would be susceptible as long as you give a certain amount of antibiotic. So if you click the link, you will go to CLSI's website where they will host the M100 document. The M100 document is the one that has interpretive criteria for all antibiotics. So it doesn't have antifungals or antivirals. It's specifically gram positive and gram negative organisms. And then this is available for free, so you can just click to enter as a guest, and then you just make sure you click on M100 document, and this gets updated annually. So for example, next year you will see the 2023 edition. So keep that in mind. And then once you go here, you can use the table of contents, and there is a table for each organism. So if you scroll down, you will see that starting with table 2A. So this one 2A is for Enterobacterales, and then the next table is for Pseudomonas. The next one is for Acinetobacter. Next one is so on and so forth. So if you are looking for breakpoints for specific organism, we're going to use Enterobacterales for this example. So if you click Enterobacterales, it will take you to the page for Enterobacterales. And this document is intended for microbiology lab staff. So a lot of this you can ignore. The part that you need when you look at the breakpoints is the table. So you will see that it has columns for susceptible, dose-dependent, intermediate, and resistant. And there are two sets of them. So one of them is based on MIC. So if you are using an automated machine that gives you MIC, or if you're using e-test, this is the part you will use. And if you're using this diffusion where the diameter of zone of inhibition is important, they will tell you, how big does the zone need to be. So in this example, let's say if you have E. coli as the organism and you're testing against ampicillin in this diffusion, they're telling you that if you measure the diameter and if at least 17 millimeter, it will be susceptible. Now, if it's between 14 and 16 millimeters, it will be intermediate. And if the zone is smaller than 13 or less, it will be resistant, including, if the zone is 0, which means that it just grew through the disc. Now, you guys don't need to memorize any of these numbers. I'm just trying to show you how to navigate and use this document. Now, with MIC, so again, for susceptible, it will say MIC of 8 or less is susceptible. So in other words, if you test it and MIC comes back and it says MIC is 4, you look at here and say, oh, good, it's 8 or less. So, for that patient, it would be susceptible. But if you had, another test result come back and said the MIC is 16, you look here and say, oh, it's 16. So it's not susceptible and it's not resistant and it's intermediate. And what intermediate means is that if you're using the usual approved doses of ampicillin, it will likely not work. But if you were to use higher doses, it will probably work. And if it's resistant, doesn't matter. If you use the maximum dose that's safe in patients, it's just not going to work. So you do not want to use ampicillin if it's resistant. And so if the MIC comes back and it's 32, it's resistant. If it's 64, it's resistant. Now, with intermediate, also in general, if something is intermediate, we try to avoid it if we have other options. So occasionally, you may not have other options available. So in situations like that, we will have no choice but to use it if it's intermediate. But if it's resistant, under no circumstances will we be using that antibiotic. In this case, susceptible dose dependent doesn't exist. So susceptible dose dependent only exists for certain antibiotics. So for example, a good example is cefepime. So if you go down to cefepime, which is cephalosporin, you will see that under the column for susceptible dose dependent, they have listed 4 to 8. So this means that if you have, for example, Klebsiella pneumoniae, which is a member of Enterobacterales, and you get an MIC for cefepime back and it's 2, you're going to say, okay, good, 2 is susceptible. Now, if it comes back and it's 8, then you're going to look here and say, oh, it's 8. It's not susceptible, but it's susceptible dose dependent. And then you can look at the notes here. So what does that mean, dose dependent? And then they will tell you that essentially, if it's susceptible, it's based on if you're using a dose of 1 gram every 12 hours. But if it's 4 or 8, you need a higher dose of higher than 1 gram every 12 hours. And they guide you to Appendix E, where they have a specific recommendation. And essentially, you will see in that part that if it's MIC of 4, you need to have 1 gram Q8 or 2 gram Q12. And if it's MIC of 8, you need to use the maximum dose, which is 2 gram Q8. Maximum, as far as FDA approved maximum dose. And of course, if it's 16, it will be resistant. And because there is a susceptible dose dependent, then there is no intermediate. So they have eliminated the intermediate for this antibiotic. So that's how you navigate the CLSI document. Here's another example. So let's say we have identified E. coli, and then we did broad microdilution to see the MIC. So, looking at this, you can see that this test tube, this is cloudy, and these 3 are clear. So, which means that organism E. coli was growing in these 2, but not here, here, or here. So the minimum concentration needed to kill E. coli in this example was 8. So MIC is 8. Now, just knowing the MIC alone doesn't tell you anything of what you can do. And I should say this is ciprofloxacin in these test tubes. So just knowing MIC is 8 doesn't mean if we can use ciprofloxacin or not. So what we need to do is to interpret this MIC. So CLSI document, I put a screenshot of the table here. You can see that for MIC, they say to be susceptible, it has to be less than or equal to 0.25, and then anything greater than or equal to 1 is resistant. In this case is 8, so it's definitely resistant. So this E. coli in this sample is resistant to ciprofloxacin. Even though, if you give high enough of ciprofloxacin, it's going to kill it, right? But it's considered resistant because we cannot given the safe doses of ciprofloxacin, we will not be able to achieve these concentrations in the human body. So we will consider resistant and not use ciprofloxacin for a patient who has this isolate. So that was phenotypic testing. Now let's take a look at genotyping testing, which is a much faster process. So these are commonly referred to as rapid diagnostic tests, also called molecular tests because they detect genes in these organisms. So there are different technologies available. One of the most common ones is a PCR, or polymerase chain reaction. A multiplex PCR, an example of it is BioFire BCID2 panel. BCID is the actual test. It stands for blood culture identification. This is the second revision of this test, or version 2, if you will. And the significance of this is that it can detect a series of organisms that's listed here. So a number of clinically important organisms that cause most common infections, including gram-positive and gram-negative bacteria, as well as certain species of fungi such as Candida and Cryptococcus species. Now, if it doesn't detect anything, it could be that there is an infection due to organisms that are not listed here, so it will not detect it. But also it is possible to have a false negative. But that is extremely rare because these tests as PCR have extremely high sensitivity. So the probability of a false negative is pretty much extremely rare, less than 1%. Now, these tests can also detect resistant genes. So there are a number of them listed here, which we will discuss in another time. Now, you don't necessarily need to memorize the list of these organisms and these genes, but what you do need to know is that this test is capable of detecting some of the most common genes for resistance. And this test is pretty fast, so you can have the results within an hour. Now, the caveat is that it will take some time for organisms to grow in the sample, and once you have a colony formation, then you can put the sample in this test. So it takes some time for those colonies to form. So it's not quite as fast as 1 hour when somebody presents to the emergency department. Another example of a PCR is a Verogen test, and it comes in 2 different panels. One is a gram-positive blood culture and one for a gram-negative. So you can see the organisms again are listed here. It works in a similar way, so it will detect genes from these organisms. So if they are detected, they are suggestive of presence of the organisms in the sample. And there are certain resistant genes that this test can detect, including some of the gram positives and some of the gram negatives. Now this is still pretty fast, so it's a rapid diagnostic test, but a little longer. So it takes about 2.5 hours once you put your sample in this machine for the results to be reported. And there is also the EPLEX BCID test that works in a similar way with similar organisms and resistant genes detected. Now, beyond PCR testing, we have other technologies. So we have MALDI-TOF. MALDI-TOF essentially can detect any bacterial and fungal organism. So it's a very powerful tool and it's pretty quick, so about 30 minutes. However, it cannot detect resistance, and there's a question mark here because it is an area of active research. So, people are researching ways to detect resistance, but clinically at the moment, this is essentially used purely to identify organisms, not necessarily detect resistance. Beyond that, we have FISH, and you can see what these abbreviations stand for, but these are different technologies. So FISH, we have Accelerate Phenotest BC that can detect a number of similar pattern of organisms, so number of gram-positive and gram-negative organisms, as well as candida species. Now this one is a bit unique because it can also report MIC, so it's not necessarily detecting a gene. So it's not the when it comes to resistance, it's not the genotypic test. It is a phenotypic test because it can report MIC for select number of antimicrobials. So it takes about 2 hours to detect the organisms, and it takes about 7 hours for these MIC results to be reported for Accelerate Phenotest. And last but not least, we have NMR and PCR. So we have a test called T2 bacteria, and this can detect Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumoniae, Pseudomonas aeruginosa, and E. coli. And at the moment, it cannot detect resistance. And this one is very fast. It takes 3 to 5 hours, but it's the time starts directly from the whole blood. So it's significantly faster than the rest of the rapid diagnostics. So if you consider when a patient comes in and we take a sample, for majority of rapid diagnostic tests, it takes some time, typically at least half a day, for this incubation to result in a number of colonies, and then those colonies go into rapid diagnostic tests. T2 bacteria is unique where you can actually take your sample directly from the blood, from the whole blood that was received from the patient without the need for incubation. This goes directly into the T2 bacteria, which significantly improves the time to identification of these organisms. However, the caveat is that, or I should say the downside is that there's only a limited number of organisms that this can detect. But as time goes on, I anticipate that this test will in time be able to detect more organisms as they research and develop this test.